free radical injury is also one of the causes for the cell injury, okay? And when there is cellular injury, there are gonna be consequences. So what is free radicals? What are free radicals? These are chemical species that have an unpaired electron in their outer orbit. It means that they are going to have unstable valencies, recalling from the chemistry, there the outer shell of the electron must be fully filled with electrons in order to be stable, but free radicals have unpaired electrons in the outer orbit. It means that these are unstable and can cause severe damage inside the cell. But there is two types of generation of free radicals. It can be physiological and it can be pathological. Physiological generation of free radicals are during the oxidative phosphorylation inside the mitochondrial inner membrane, okay? And all of the human beings and living organisms rely on oxidative phosphorylation for the generation of ATP. So in oxidative phosphorylation, there is an enzyme known as cytochrome C oxidase. To be more specific, it's complex 4. It transfers electron to oxygen. Okay, and when the oxygen is reduced, you know that electron transferring, it means that one atom accepts the electron, it's called the reduction. And when it yields the electrons or give away the electrons, it's known as oxidation. So when the oxygen is accepting the electron, it means that oxygen is undergoing reduction process. When oxygen accepts one electron, it forms superoxide here, and when it accepts two electrons, it forms hydrogen peroxide, and when it accepts three electrons, it forms hydroxyl radical, and when it accepts four electrons, it forms water. It means that till here it's complete reduction, but till the hydroxyl radical it's partial reduction, and partial reduction of oxygen is going to yield the radicals which are free radicals, okay? Oxygen is completely reduced to water, but when it's partially reduced, it's gonna yield superoxide, hydrogen peroxide, and hydroxyl radicals in its way. This was physiological generation, but what about pathological generation? Pathological generation is due to the ionizing radiations. It can be gamma rays, X rays, okay, it can be UV rays as well. So the ionizing radiations acts upon the water and it converts the water into hydroxyl radical. And recalling you that hydroxyl radical is one of the most dangerous free radicals among the all, and it's most damaging to the cell. Ionizing radiations converts water to hydroxyl radical. Also, the second pathological generation is about the inflammation. You know that in inflammation there are a lot of cells, the white blood cells and those cells which are causing damage to the system. These cells must be removed by the white blood cells, and white blood cells uses two mechanisms to kill those cells. These are oxygen dependent killing and the second is oxygen independent killing. In oxygen dependent killing by the neutrophils is a white blood cell, it utilizes the oxygen and converts it into superoxide by the enzyme NADPH oxidase, and NADPH oxidase is used in the oxygen dependent killing by the neutrophils in inflammation. When oxygen is converted to superoxide, superoxide is a free radical, okay? So it's the pathological generation of free radicals during the inflammation. And the enzyme used is the NADPH oxidase. The third is the metals. You know that there are several metals, metal atoms inside the body. For example, copper, it can be iron. and these are normally in the bound state. They must not be present in the free state. But when these atoms or metals are present in the free state, they can cause damage. They are normally in the bound state. Either they are bound to the metal carrier proteins or they are bound to plasma proteins. There is a very high yield reaction known as the Fenton's reaction. Okay, in Fenton's reaction, it's related to the iron, in which the iron is used to produce the free radicals. Iron is in two forms. It's in ferrous form and the second is the ferric form. The ferrous form is the 2 plus form of the iron, okay? And the ferric form is the 3 plus form of the iron. When the ferrous, which is 2 plus form, combines with the hydrogen peroxide, it's also a free radical. It is converted into ferric, the 3 plus form, and it yields the most dangerous hydroxyl radical, which is known as the hydroxyl radical, and it's the most damaging among the all. This is the Fenton's reaction. To be more specific, it's high yield. There are several diseases inside the body in which these metals can build up inside the cells or inside our bloodstream. For example, the first is the hemochromatosis, and as the name suggests that it's heme, it means that the iron builds up inside the body. Its pathophysiological mechanisms are we are not going to discuss in this lecture, but you just need to remember that this disease causes the iron to build up inside the body. And the second is the Wilson's disease. In Wilson's disease, the copper is excess inside the body. So these two diseases can cause free radical damage to the cells and the system. Okay, drugs and chemicals. The drugs and chemicals can also generate free radicals because these drugs are metabolized by the P450 system of the liver. It's the cytochrome P450 enzymes that are present inside the hepatocytes, and these metabolize several drugs and chemicals. Drugs, for example, acetaminophen, and chemicals, for example, carbon tetrachloride, which is CCl4, and these are metabolized by the P450 system of the liver, and during the metabolism they can generate the free radicals. In During the generation of the free radicals, they are going to metabolize the drugs. What are the damages that is caused by the free radicals? How the free radicals is going to produce harm to the cell or to the body? First of all, the free radicals brings up about two responses. First is the peroxidation of lipids, and the second is the oxidation of DNA and proteins. In the peroxidation of lipids, as you know that cell membrane is formed of lipid bilayer. It's formed of lipid membrane. So when there is peroxidation of lipids, the cell membrane is going to get damaged. And in the oxidation of DNA and proteins, the cytoskeleton of the cell, it's made up of proteins, and DNA, which is present inside the nucleus of the cell. It's most important for the structural and functional stability of the cell. So when its oxidation is going to occur, the cell is going to suffer, and when the DNA damage occurring, it results in aging and oncogenesis. Aging is irreversible process in which the cell is going to age irrespective of what we use, and oncogenesis is the production of the cancerous cells. So having discussed this, we have to also discuss about the elimination of free radicals. Are we only generating the free radicals, or there are any means that free radicals can be eliminated from the body? Yes, luckily there are means or enzymes that can cause the elimination of the free radicals. First is the antioxidants. Antioxidants includes vitamin A, C, E, and glutathione. As you know that many cosmetic doctors suggest IV glutathione drips so that it can bring about antioxidant effects. So the second is the metal carrier proteins. As I have discussed that the metals, which is iron and copper, when they are in free state, they are not bound to something. They can cause the damage. But when they are bound to some sort of proteins or metal carrier proteins, they are in bound state, so they produce less damaging effects. So the protein which is binding to the iron is the ferritin, okay, and the protein that binds to the copper is ceruloplasmin, and these are of high yield because these must be present inside the cell as to bind these metals. The third mechanism of elimination is the enzymes, the presence of enzymes. As I've told you, that reaction is oxygen accepting one electron converts into superoxide. It accepts two electrons and converts into hydrogen peroxide. And when it accepts three electrons, it's converted into hydroxyl radical. There are enzymes that act on several steps in these reactions as to eliminate the subsequent free radical. First is the superoxide dismutase. Superoxide dismutase act on the superoxide, and when it act on the superoxide, it converts into hydrogen peroxide. It eliminates the superoxide, right? Okay, and catalase is an enzyme that acts on the hydroxyl radical, and it converts into oxygen and water. So it's going to eliminate the hydrogen peroxide in the reaction. And glutathione peroxidase is a very important enzyme because it's gonna bind to the free radical, and it's going to convert the free radical into glutathione disulfide and water. Okay, these three enzymes, in the way of their reacting mechanisms, they eliminate the free radical and convert it into other substance or atoms, and they eliminate that free radicals. Okay, glutathione peroxidase combines with the free radical, converts into glutathione disulfide and water. So this reaction is a main theme, so you can acted by several enzymes in this series, and can convert into water and oxygen and hydrogen peroxide. Okay, free radical injury. Free radical injury. First of all is the we are going to discuss about carbon tetrachloride in detail, and the second is the reperfusion injury. Carbon tetrachloride, as I have told you, that it's a chemical and it's used in the dry cleaning industries. For example, a clinical vignette shows you that a person works in a dry cleaning industry, so you can suspect that he has been exposed to carbon tetrachloride. And as I have told you that cytochrome P450 system of the hepatocytes act on these chemicals, and when these act on these chemicals, it converts CCl4 to CCl3 by the P450 system of hepatocytes. But CCl3 is a free radical. Okay, CCl3 is a free radical. So it's going to injure the cell, and during the cellular injury, the hallmark for the injury is the cellular swelling. And when the cellular swelling occurs, the organelles inside the cell also swells up. For example, rough endoplasmic reticulum. Rough endoplasmic reticulum swells up, and as you know that on the surface of RER, there are ribosomes attached. And ribosomes due to swelling gets detached, and when detachment of the ribosomes occur, there is reduced protein synthesis. As you know that ribosomes are the factories for protein synthesis. When there is reduction of proteins, what is the consequence? It results in decreased apolipoproteins. Apolipoproteins are the proteins that bind and transport the lipids. Okay, these are apolipoproteins. But when there is decreased apolipoproteins, it leads to the fatty changes in the liver, because the liver is not able to bind the fats and transport it out of the liver. So liver So fats and fatty acids and cholesterol and other sort of fatty substances builds up inside the cell, inside the liver cells, so it results in the fatty liver changes. The second is the reperfusion injury. Reperfusion injury means that when there is already ischemia, okay, to a particular tissue, and there is return of blood to that ischemic tissue. But why it's damaging? Because in the returning of the blood will bring oxygen and other inflammatory cells along with it. And these oxygen and inflammatory cells are responsible for the production of free radicals in that ischemic site. So it's going to damage more. Paradoxically, we would suspect that return of the blood would result in healing, but the situation is opposite to that. The returning of blood to already ischemic tissue would result in the production of free radicals, and there is more damage to that area or to that tissue. So this was for today, and we will see other forms of injuries and in the next lecture. So till then, goodbye.